Okay. Good evening. This is Dr. Shen again from West Virginia University. So we will continue to talk about genetics. Today we will talk about RNA transcription. So let's still go back, talk about this central dogma. Crick mentioned the central dogma back in 1950s. This is DNA goes to RNA, and RNA goes to protein. And we already talked about the DNA replication. And we mentioned that this is always 5' to 3', rely on DNA polymerase. And today, we will talk DNA goes to RNA. This is called transcription. And the next video, we will talk RNA to protein. This is called translation. Now, before we talk about anything, we had a very simple question I want to ask you. How are you understanding why we call it transcription? And also why we call it translation. So first of all, what is transcription? You know that this is letters to letters. So what are they? It's a code of DNA to RNA. And most of the time we talk about mRNA. Now, how about translation? This is we call letters to protein. So do you feel about the difference? This is kind of like I speak Chinese, and if I don't understand the English, somebody will do the translation for me from Chinese to English. It's the same here. Now, another thing I want to mention, we're going to go talk about DNA, RNA structure a little bit. Those are very boring chemicals. And compared to protein, you have amino acids, you have peptides, you have a protein structure, you have a primary, secondary, tertiary, and the quaternary structure is more prolific. So you understand this is also from a boring structure to a prolific life. FIC. So this is also part of the meaning for translation. Okay? So today we're going to focus on talk about this transcription. Now before we're going to go detail of the transcription, we want to mention a little bit about the difference between DNA versus RNA. Okay, so we want to talk about that one first. First of all, let's still review what is DNA. 2' deoxyribose nucleic acids. Then what is RNA? And I'm going to remove this RNA a little bit far away because I could make sure you know the difference. Okay, what is RNA? Deoxyribose. If the first time learner, I would suggest you just say oxyribose nucleic acids. Now, of course, we don't have to mention this. Therefore, you can take it out. Now, let's draw the structure. Okay, what it looks like. 5 carbon. We call it pentose. Okay? Here, we're going to attack to a base. In H, we're going to have H, 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 OH, then go OH, CH2. Then here, never forget we had a beautiful phosphate cell. Now the big difference is here. So if we do the label, this is 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime carbon. Here, we do not have an O. Okay, ribose here, you can do the same thing. What's the difference? Don't ever hesitate by yourself to draw it, because that will make your life more easy and more understanding. Okay, H, OH, CH2, OH, and goes on the top. We always say you have oxygen and a phosphates and a phosphates. Okay, right here, don't forget, you had the O there. And I want to mention this difference. We want to later on talk. When you see the OH, the hydroxy base, you kind of feeling this base gonna react with something. Okay, so that's I'm gonna talk right now. This is the first difference. Second, you know there is a base connected to that. Okay, what are the base of DNA? A, T, G, C. These are the bases. So adenine, thymine, guanine, and the cytosine. How about the RNA? What is going to be the RNA looks like? We still have A. Do we have T? No, we don't have T. We have U. We still have G and C. So, instead of we have a T, we had a U. What is U? Uracil. The uracil replaces thymine in the RNA structure, which is at the base. Now, by the way, if you look at the detail of the structure, this is we call beta nucleosidic bonds, which goes up. If something goes down, we call it alpha. However, alpha does not exist in the natural environment, so we do not mention this one at all. So therefore, we skip to talk about these details. Okay? Number 3. We mentioned for DNA, deoxyribose right here. For RNA, this O is there. And this is not very stable. Give you a feeling there's something going to happen for the reaction. And look at the base. You have T in DNA, U for RNA. And I also want to mention that A complement with U is always not going to hold on something very easy. So we can say the DNA is relatively stable and the RNA relatively unstable. Last, I want to mention, everybody knows, for DNA, most of the time is double strand. And you know that, antiparallel double helix. For RNA, most of the time is single strand. Of course, we know there is some exception. For environmental bacteria, for virus, for archaea, sometimes there is some exception. So these are the things. We talk about the difference between DNA and RNA before we talk anything. Now, by the way, remember when we study, we need to connect the knowledge from different sections. What are the structure here? That's called the pentose. Where they comes from? Pentose phosphate pathway, if you still remember. And what are the reducing energies they generate? Is NADPH, nicotinic adenine dinucleotide phosphates. Okay, so these are the things we mentioned the difference. When you look at the structure, are you feeling they're so boring? Ribose, base, phosphate. That's all. Compared to the protein, this is so boring. And the protein is more prolific. Okay, so this is the first thing we talk about, is the difference between DNA and RNA. Now, the second thing we want to mention, since we talk about RNA, so we're going to mention a little bit of detail about RNA. So first question for you. What are the three major differences, major types of RNA? Number one, mRNA. We call it messaging or message RNA. Why? Because they have a genetic code. Number two is tRNA. We call it transfer RNA. Why we call it a transfer RNA? Because they carry on anti-code. Be careful. This in the exam always going to ask you. They are carry on anti-code, not the code. Okay, what is the tRNA looks like? Do you remember? How about let's draw a basic structure? What it looks like? Do you feel it's like a tree leaf? Goes here, this rise a little bit higher. And these are the places which carry on is called anti-code arm. And this is we call T theta C arm. Now here is a very simple question, sometimes in the exam. If I mix mRNA and tRNA, by the way, there is another one we call ribosome RNA or RNA. How do you know it is tRNA? Because if we do the isolation, we find that tRNA carry on some of the gene bases very weird. So there's some of the weird gene base. What are they? What are the examples? T, theta C, and PU. Those are weird genes. If you see weird genes base, that means it's possibly is tRNA. Okay, what is ribosome RNA? What is rRNA? R means ribosome. Okay, we want to talk about this. What is it going to look like? One unit, another unit. Big units, we call it 50S. Small units is 30S. They add up together is 70S. Now I'm talking about the example is a bacteria. I told my students, never take it for granted. These two numbers is not add up together. It's 70S. Why? This S is Svanberg units, which means it's measured by the precipitation speeds after centrifuge. It is not a molecular weight. So don't take it for granted it's going to add up. It's not. Okay, what's the detail of that? In 50S, large subunits, you have 5S and 23S, all RNA. And these responsible for transpeptides after the transcription during translation. How about 36 subunits? We have 68 rRNA. This is a very magic stuff. In 68 RNA, we have a very conservative sequence, V8 and V6. V6 to V8. There is a conservative sequence universal for all the bacteria. They can generate some of the primer, can test all the bacteria. That's why we call it 16S rRNA molecular technology. And remember, Dr. Wei-Yun Zhu in Animal Science and Technology Department has a tool which is called 16S rRNA DGGE. This is where its original come from. And I still remember I had my first published paper in peer-reviewed journal of Applied Microbiology back 2007. I did the research 2004, about 17 years ago. This is what's the story about that. Okay, so we finished talking about the three major components, major types of RNA. Then we're gonna move on to what we called transcription. Okay, so I'm gonna move everything, go to the side of the corner. Then we'll talk about the detail. So we want to mention about the transcription. The detail. Now, when we talk about the transcription, we will be focusing on three steps. Initiation, number one. Number two, elongation. Number three, termination. And we're going to say initiation is more important. Okay, so let's talk about it. So first of all, let's talk about initiation. So first of all, you need to know what happens on the initiation. Say it very simple. Is DNA promoter combined with a remarkable enzyme called RNA polymerase. And we're going to get on that. So let's talk about the promoter first. Okay, what's that? Let's do something easy. In an E. coli cell, we use precaryous as an example. So in an E. coli cell, let's say we have a double bond. Okay, right here. 3' to 5'. And 5' to 3'. I may need to make it a little bit large because we're going to write something there. So we're going to draw a little bit large. That's too small. So this is 3'. Let's say our starting place. I had a gene base is A, complementary with T. This is called +1. Now, I already said it before. Anything on the right side, this is called downstream. Anything happened on the left side, so I'll just put it in there, called upstream. This is the first direction you need to know. Now, for the DNA promoter, they have a conservative sequence. That's why the RNA polymerase can recognize that. What are they? Above 10 BP base pair, in the upstream of the starting points, there is a first conservative sequence called TA, AT. No, you can do a corresponding. It is AT, TA. This very conservative sequence is called -10 sequence. We gave them a name called pyramidal box. So this is -10. There is a little bit of space between that. It is about 16 to 18 BP base pair distance. We have another very conservative sequence. Is TT, GA, CA. Now corresponding to the other bands is AA, CT, GT. This is called -35 sequence. These two very conservative sequences, if we put them together, cut it, this whole area is called DNA promoter. Now this DNA promoter gonna be recognized by this chunk, a large chunk. And this large chunk is RNA polymerase. Now let's talk about what is RNA polymerase. Okay, first of all, RNA polymerase, I said it's remarkable because it has lots of subunits, lots of components. What are they? It is composed by alpha 2 subunits, beta, beta prime, and omega. What is alpha? This base pair about the subunits is about 36,000 DA, delta units, molecular weight. This is the subunit. Basically is responsible for unwound DNA and assist for RNA synthesis. Beta and beta prime, those molecular weights are much larger than alpha units. What is the molecular weight? Beta subunit around 150 kDa. And the beta prime is the largest one. It is about 155 kDa. These two are responsible for RNA synthesis. Now always remember the direction is 5' to 3'. Same as replication, DNA replication. How about the omega? Omega, this is the smallest one from the molecular standpoint. And I will say the function is not really clear. We call it unknown sometimes. But people will say it is assist for assembling RNA polymerase with DNA promoter. Now, don't forget, there is another magic stuff there, is sigma factor. And most of the time, regarding a bacteria, for the universal life function of a bacteria, this sigma factor is sigma 70. Why we call it a sigma 70? Because it's 70,000 dA. Sigma factor is help the DNA polymerase to recognize the very conservative DNA, to help RNA polymerase to recognize the DNA per motor. Now, one more thing we want to mention. These 4 subunits, 4 different types of subunits, actually is 5 subunits because we have 2 for alpha. This is we call core enzyme. And the sigma factor, we call it cofactor. When you combine them, we call it a hollow enzyme. Do you still remember when we talk about a hollow enzyme? What is that? I'll just remove this part, then we can talk. We do a little bit of review, okay? So we have a we have an apple enzyme. And sometimes we call it core enzyme. Then we have like a coenzyme. This combined, we call it a hollow enzyme. Now the coenzyme also can separate into 2. If it's loosely attached, we call it cofactor, like sigma factor, because it's gonna moving. How about if it's closely attached? When it's more stuck there, this is called prosthetic group. Okay? So this is sigma factor. Now, I also want to mention, when we talk about this sigma 70, we always say this sigma 70 is for major and diverse function of a bacterial cell. In the stress environments, bacteria has their special RNA polymerase to survive. Let's say in a thermal inactivation status, and we're gonna have a heat shock protein. Now, at the end of the day, bacteria has to translate it as a heat shock protein to survive. So during the transcription, that is not sigma 70, that is sigma 28. The sequence is different. Now, how about a very important for bacteria has chemotaxis and the flagellar components, and also for real motility. This will be responsible is sigma 32. Now I still remember the sigma 32, the conservative sequence, is TAA, about 15 BP base pair distance. Then you have a GCCGATAA. That is going to be the per motor. The conservative sequence, the sigma 38 will be recognized. They will not recognize this one. So I just want to let you know, in your textbook, when we mention about sigma 70, is a universal sigma factor to help RNA polymerase to recognize the DNA per motor. But for bacteria during their life cycle, it is actually have multiple different sigma factor to recognizing a different situation. Talk about motility. Do you still remember? It is important. Talk about the flagellar components. Do you remember a theory called chemomosis? Who mentioned about that? Peter Mitchell. In 1930s, he won the Nobel Prize in 1960. What is this about? Do you remember it? Let's do some of the review. So we have NAD go to FADH2, NADH2. Electron go to FES, FMN, and then go to coenzyme Q, then go to cytochroma C, A3, and A. So when the electron is transferred during the electron transport chain, what's going to happen? Something going to go out of the cell membrane system. That is protons. Okay, so protons accumulated right here. This is called proton motive force. When this proton motive force, they will dig a hole here, go inside, trigger ATP synthesis. Now what's going to happen? ADP with phosphates becomes ATP. At the same time, if bacteria has a flagella, it's going to trigger with flagellar rotation. Okay, let's say it's very long if it's like this. This flagellar rotation is responsible by sigma 32. Okay? This is a series important because that's back for oxidative phosphorylation. So just want to do a brief review. Let you know, not always is sigma 70. Although the textbook mentioned, it's just a model. In a growth situation, bacteria have a different sigma factor to recognize the different DNA promoter. Okay, so this is we finished talking about the initiation. Then we want to talk about the elongation. What's going to happen for the elongation? So we're going to remove this one because this is not related. I'm just talk something else and try to help you to understanding the different situations. So we're going to remove this part. Okay. Sigma 70 also stay. Okay, now let's talk about elongation. Second, elongation. Okay, what's going to be the elongation looks like? Now let's draw a beautiful picture. Okay, one strand of DNA. Okay, let's say 3' to 5'. Another one, 5' to 3'. Okay, what's going to happen here? They separated. They need an RNA polymerase to recognize the promoter, and they need help. Is sigma factor. Where going to be the RNA comes out? So RNA will come out this way, 5' to 3'. So, at this moment, we will say number one. They are forming a DNA RNA hybrid bubble. Now, what this means, if I take this part goes here, you will see more clearly. I have a 3 prime, 5 prime to 3 prime. Another DNA bands running through 3 prime to 5 prime. The top one we call it a coding bands. And the bottom one we call it a template bands. Template band, I should say. Now, why we say it's a template band? Because they're based on gene complementary policy. They're going to have the mRNA comes out. So let's do some review. If we have A, T, G, C, C, let's just say short 5 base pair. Here we're going to have T, A, C, G, G. What is going to be this mRNA looks like? This mRNA should be exactly the same as the coding band. But the only thing is this is not going to be T, it will be U. So this will be A, U, G, C, C. Be careful of the trick. 5 prime and 3 prime. So this is if we are isolated from there, we will see what happens. Okay? So this is, we talk about is elongation. Now, there is something we want to mention. It is a major difference compared to the DNA replication. RNA polymerase is remarkable. It can unwind the DNA. It will moving through the double strand of the DNA. It will biosynthesis RNA. However, there is no error fixing function. So what is going to happen? If there is some mistake right here in the DNA replication, they will be carried to mRNA. At the end of the day, will become a protein. It could cause molecular disease. That's what you mentioned in your textbook. I just want to make sure there is no error fixing function here. It's very unfortunate. Okay, there is one thing I want to mention. We mentioned RNA polymerase here. Who found this? They're spending another almost 70 years they do the work. Even today, you see people still do the structure of RNA polymerase. And I tell you one thing, in 1960, the Nobel Prize was awarded to 3 American scientists for RNA polymerase. However, it's very embarrassing. 10 years later, they find it is not really RNA polymerase. It's some of the mutations from some archaea. In 2006, Roger Cronenberg got a chemistry award of Nobel Prize because he is the first person to picturing RNA polymerase. Now my question is, did you listen to my talk very carefully? Who's that? Arthur Cronenberg's son. Arthur Cronenberg is the person who found DNA polymerase. Okay, so their family, they are doing great work to talk about the central dogmer, DNA polymerase, and the RNA polymerase. Okay, so we finished talking about the elongation. We're going to move on to the third stage, which is termination. So I have to remove this, because you already know central dogma very clearly, believe me. Okay, what we're going to move to the section is called the termination. Okay, when we talk about termination, we're still going to draw this. And I tell you one thing, don't hesitate to draw this on your notebook. Because it's always help you to understanding these materials. Okay? What's going to be the termination? So let's say we have the RNA, mRNA biosynthesis goes here. Okay? What's going to happen? There is a two basically mechanism. Number one, we call it rich U region. What happened? When they started to biosynthesis, okay, let's say here is small segments of the RNA nucleotides. They added. There is a one stage. They could become U. And the corresponding become A. Now this is DNA. This is RNA. They corresponding with each other, is that right? But I already tell you right here, if you still remember, the rich U area, the AU combination, is very weak bonds. You can go back to look at the energy, the delta G. This is very weak. It's not gonna hold on too long. So therefore, they stopped. What are they gonna stop? At the end of the day, something like this is gonna generate. This is called stem loop hair spin structure. And at the end of this is going to be U, 6U. Okay. This is why we usually call it internal. So since we have internal, we're going to have external, is that right? What's going to be the second one? Rho factor initiated terminator. This is, we call it external factors. Okay, what's going to happen? Instead of, I'll just draw here. Now you have this 5' to 3', mRNA comes out. This chain going to become very, very long. You're going to have something area called RUT sequence. Once this RUT comes out, what's going to happen? This Rho factor will be initiated. And then they start to generate some of the chemical like this. This is a terminator. Once the terminator happens, they also gonna happen created a same stem loop hairspin structure. Then they stopped transcription. So the same thing here. At the end of the day, these two functions is all come out with a stem loop hair spin structure. Okay? So this is, we finished talking about initiation, elongation, and termination. Now, you're going to ask me, Dr. Shen, you talk about E. coli. I'm more interested about other things. I like yeast and modes. For you guys, you like animals and the plant cells, which means they are eukaryotes. Is eukaryotes going to be the same thing like this? That's going to be your work to do. I'm not going to teach you everything, is that right? So what you going to do? Go home on your notebook. You're going to review all this stuff. Then read your textbook. I believe your textbook have it. Do the comparison. What is the eukaryotes and the prokaryotes difference during the transcription? But since I'm here, I'm going to give you some of the highlights, which means some of the hints for you. The difference between eukaryotes and the prokaryotes. So we want to talk about some of the highlights of, for me, is yeast and fungi, RNA transcription. Okay, what are the highlights then? Number one, as you know, the eukaryotes, their function is more divided, more classified, which means more individual. So, their RNA polymerase will have individual function. RNA polymerase 1, responsible for transcription, ribosome RNA. RNA polymerase 2 is same we talked about here, transcription messaging, mRNA. And RNA polymerase 3 responsible for transfer RNA. This is the first difference, is more classified, more individual function. Number two, look at the promoter. It's very specific, is that right? Conservative sequence. For prokaryotes, is also going to be more conservative, different. They will have TA box as their promoter sequence. They will have so-called CAAT and the GCCAAT box. Now, very similar to the prokaryotes. Those two also located in the upstream. This is about TA box is about 30 BP base pair upstream. What I mean the upstream is compared to the initiator place, which is +1. GCCAAT box is about 50 to 100 BP upstream.